All right. Everyone, welcome to 2021's 3-minute thesis competition. Sorry for a couple minutes there as we set up some of our features. So we're doing this virtually for the first time, but this is the fourth time that we're doing NMU's 3-minute thesis. So it's new and old hat at this point. Thank you all for being here. Thank you for supporting our student researchers. And I think it's going to be a fun event. We're going to all learn a lot. So first up, I'll tell you a little bit about 3-minute thesis. So 3-minute thesis was started by the University of Queensland in Australia. Their idea was that thesis defenses and research can get very long to talk about. So they came up with a format where it's very short, it's concise, and also a way to convey it to a general audience. So that's what we're all doing here. We're going to learn about some research in a very quick format and in a way that everyone can understand. So it's actually kind of interesting. They came up with this during a drought in Australia. So they all had to take 3-minute showers. And their dean of their graduate school was taking one of these 3-minute showers and was like, you know what we should do? Present in this amount of time. So that's where it started, of all things, in 2008. And then it's actually spread across the world. It's now in 900 universities in 85 different countries, which I think is one of the coolest features about 3MT. We're part of a global research network, and that's really awesome. And it's something that we've seen certainly now as we're doing virtual. They're doing it across the globe as well. So we've all come together to figure out how to do this. And thank you all for still supporting things. Even though we're not in person, we're still going to have a good time. And we've got as much of the original fun from the normal format as we can do in here. And hopefully it's a good experience all around. So with that, I will turn it over to the Dean of Graduate Studies, Dr. Lisa Eckert.
Welcome. I will share my screen with you so that you can see me. This is a little, go to main, there we go. I'm Lisa Eckert. As Janelle introduced me, I'm the Dean of Graduate Studies and Research here at Northern, and I am honored that you have joined us in this event. This is my favorite event of the year because it's all about the students, and the students do the talking and they tell us about their research and share some highlights with us. They show off their knowledge and creativity, and we learn something along the way. This event highlights the synergy between research and education and the magic that can happen when discovery and creativity combine into that aha moment of clarity. This event also showcases NMU's commitment to undergraduate and graduate research and creative scholarship. As you know, we have McNair students who are coming first. Those are the undergrads, and then we'll be moving on to the graduate student round. They will share their work with you from their graduate capstone projects. They come from programs across campus, and we get to see that they see the light at the end of the tunnel, and they get to share all of the work that they've done, and you will help us celebrate each and every one of them. I'd also like to thank the volunteer judges who are generously donating their time in support of our students. Janelle will be formally introducing you to them in a few minutes, but I also wanted to express my sincere appreciation for the gift of your time. We can't be successful also without the hard work of a team to put an event like this one together. So thanks primarily to Janelle for moving mountains and making this virtual event possible. I would also like to thank Jeff Koval from AV, who patiently answered our questions as we prepared for this event, Angela Rasmussen, Felicia Hokenstad, Adam Klingspan for all of the work they did in preparation. Also, Lee Chang, who you will be meeting in just a few minutes, Lindsay Huckala, and Heather Pickett. And hopefully I haven't left anybody out, but there's a lot of people who are behind this, and thanks to each and every one of them. And I'm not going to take up any more of your time because that's not what you're here to see. So thank you one more time for coming, and I will enjoy the show along with you.
Mute. Thank you for those who said I'm on mute. So again, sorry, my name is Lee. I'm the director of the McNair Scholars program. Thank you, Lisa, for the introduction. I'm just here to tell you a little bit about the McNair Scholars program and why we are also part of 3MT. So the McNair Scholars program is a federally funded TRIO program. We are one of 6 programs in all of Michigan. And in the United States alone, there's 151 program. And I thought it would be really cool to share this statistic with you because this program, unlike any other TRIO program, specifically helps students from low income first generation or students from underrepresented ethnic and racial minority groups to get into graduate school and hopefully to get their PhD. So research is a big part of what we do. And we do help fund our students so that they can get faculty guided research. They also get stipends in the summer so that they can participate in research as well. So some of our students are here today participating in this competition, not in the traditional competition, but in their own way in the competition that we've created that is happening along the side with 3MT. And I also wanted to just take this moment also to plug in that the winners of 3MT will also be sponsored or will be, their videos will all be made available during our annual celebration of student scholarship. This year we're celebrating the 25th year of this celebration event. Basically what it is an event to celebrate students and the scholarship and research creativity that they've been doing for the last year. We're going virtual this year. We're going to be in a virtual world. As you can see from the flyer, the virtual world is right there. So if you have nothing to do, or if you want to look at some more scholarship and some more creativity and work, please join us on April 7th through the 8th as we celebrate student work. And I'm going to hand it over to Janelle from here.
Thank you, Dean Eckert and Director Lee. All right. Thank you, everyone. I'm back on. There's a lot of button clicking on this end, so please bear with us. I think I actually have a slide about that. Please bear with us as we switch back and forth through the whole event. So I'm here back again to tell you a little bit more about 3MT. So you can see here, these are the rules. These are what the participants will hold to. The main point of it is that you have a one slide and you have 3 minutes to talk. Basically, there's no media, there's no instruments, there's no costumes. It's trying to make it kind of a level playing field for different researchers to all be able to talk really about what their research is. That being said, we do have judges who will be doing the final decision, who wins. So I'd like to take this time to introduce to you our judges. Unfortunately, Kurt was unable to make it here today, but we do have the other 4 judges, and they are here with us. So if we can, judges, if you can turn on your mics and your cameras, you can introduce yourselves briefly to the rest of us, and that'd be great. I'll stop sharing for that. Oops.
Would you like us to go in a certain order, Janelle?
Oh, sorry. If you want to go alphabetically by last name, that's fine, just because that's how the slide is. If you want to jump in and talk over each other, that's fine too.
All right, I'll go first. Hi, everybody. I'm Deb Brunell. I am the Director of Workforce Services for UP Michigan Works, and I'm happy to be here and get this opportunity to be with all of you. Thank you.
If we're going alphabetically, I think it's my turn. My name is Nathan Larson, and I'm a 2-time grad of Northern, and I'm so excited. You know, the titles of all these presentations are so exciting. And I work as a nutrition teacher for Marisa, and I'll hand it over to Kristen.
I want to say a big hello to all my fellow Wildcats out there. My name is Kristen Lucas. I'm a 1995 graduate of Northern Michigan University and am now Associate Dean of the College of Business at University of Louisville. And I'm just so thrilled to be hearing about all of your research.
And I'm Chris Standerford, also an NMU alum, currently the director of the Seaboard Math and Science Center. And super excited to be here. I consider myself a lifelong learner, and the research that all of you have done, again, I have goosebumps already. Super excited to hear all the things that you've discovered. So thank you.
And we do want to say a big thank you to all of our judges. You guys are amazing for donating your time and your energy and your expertise to us and to the students. So thank you very much for being here. And I'll bring us back up here. Let's see. All right. So this is a quick overview of what our judges will be using to judge the participants. This is commonly available out there. It's the same criteria that's used worldwide. The main point is that participants should be engaging the audience. It should be understandable. They should bring you kind of into their research with their slide and the way they talk, and also that their research itself is kind of engaging, it's in depth, it makes sense, it's something that you're excited about. These are important because our judges aren't the only judges in 3MT. You're also the judge. So everyone who's attending right now, everyone who's live, there will be, there's a category called a people's choice. So the judging panel will decide the winner and then the runner-up in both categories. And then in both of those categories as well, there's a people's choice. So at the end of when the last McNair speaker speaks, and then when the last graduate student speaks, we will come up, there will be a poll on your screen, and it will ask you who you want to vote for your people's choice winner. So you as the audience need to pay attention and decide what you think who you think is doing kind of the best. You can do it on your own criteria. You can use the judging criteria. But just be aware that'll show up. You'll have a few minutes to decide, put those in, and then that person will be announced along with the winners at the end of the contest today. Also, since this is virtual, there's all kinds of little nuts and bolts that we all have to talk about. Just know that we're doing a Q&A function at the end. So normally we would just have an audience kind of raise their hand and ask a question of the researchers when they're done. In this format, we do have a Q&A option in your Zoom. So you can click on that at any time and enter in a question. Lindsay Huckla is collecting those, and she will go through those. And at the end, when everyone has already spoken and the judges and the people's choice votes are in, we will do a Q&A so you can learn a little bit about the students' research that maybe they don't cover in the 3 minutes. You can ask what they're doing. If we can, we'll keep it pretty fun and not too technical, just kind of some general questions about their research. And then we'll let a few people answer some of those questions for the audience, talk a little bit more about themselves while we do some of the behind the scenes adding and judging and that kind of thing. But I do want to stress it's not part of the presentation itself, and it shouldn't be part of anyone's judging or choices. That's just kind of an add-on fun thing to learn a little bit more than you can learn in the 3 minutes. Also, there's that bit, we will have some transitions, as you've already noticed. So use that downtime to think about any Q&As, any questions that you want to answer, and your people's choice decisions as well. So with that, we'll get started. So our first competitor today is Jalen Sims. He's a McNair Scholar, and he is in the graphic design department. And he'll be presenting alternative materials of architecture. So I will switch over to Jalen, and he can start when he's ready.
Okay, hello everybody. Like Janelle said, my name is Jalen Sims. I'm a graphic design major, and I'm part of the McNair Scholars program. So for my research project, I will be sharing with all of you a study on urban architectural design and how it can be better integrated with nature and sustainability. The biggest thing about this project was I wanted it to be consumer friendly and easy to understand, because the people that are going to be hopefully using these are people just like you, who are not like major construction people and you're just average citizens. So starting at the top of this slide, I wanted to focus on ways that we can reach symbiosis with our environment. So the first thing is water. Some of the ways that we can better integrate ourselves with this abundant resource is by using pond or a water tower as irrigation for the plants in our area. Another thing we should be doing is investing in hydroelectricity because this is a great way to get energy that we need. And finally, we should think about using vertical hydroponic systems as a way to grow food because they use less water and soil and they're more productive than traditional farming. Moving on to plant life in our area, we should be planting native grasses and soils on our roofs to attract birds and also insulate our homes better. We should also have vines and ivy hanging from our roof to help offset our carbon input, and that can also be a great natural decoration for our space. Next up is wind energy. We should be investing in wind farms, and when it comes to buildings, we need to make sure that all windows are able to be opened so we can better regulate temperature without having to turn the air conditioning on. And finally, there's solar power. We should be investing in solar panels on our homes and around our cities as the way to get energy. We should also focus on building designs that have lots of windows in them to allow natural light to come in, which boosts people's mood and saves on electricity. Looking at the bottom left portion of the slide, I have found 6 different materials that are an alternative to concrete and other standard building materials. So the first one is straw bales. These are great for insulating a home and they're a rapidly renewable resource. Hempcrete is very easy to make. It's stronger than concrete. It's carbon neutral. And it's actually great for farmers because the root structure, it actually breaks up the dirt in the soil, allowing the next crop rotation to easily get nutrients. Recycled plastic or eco bricks are great because it finds a new use for all the plastic we're creating and can be very cost effective, and especially in areas that have less money and less resources. And before my time ends, I wanted to take a moment and talk about the list over on the right. I encourage you all to take a picture of this list as it can be a way for you to keep track of, or by doing the things on this list, you can increase sustainability of your home and your life. So a green roof is when you have soils and grasses on your rooftop to insulate your home. When working on construction projects, we should keep in mind these alternative materials that I've showed you. And yeah, please take a picture. And that is my 3-minute thesis.
Thank you very much, Jalen. All right, so we will let the judges kind of mark things down. Everyone can think about what they've just learned about these things, and I'll bring up our next slide.
Hi, I'm one of the judges. Can we unmute for a second and clap for the presenter?
Sure.
Okay.
I don't know. I think only the judges and the participants can clap, but we can all clap.
Okay. Thank you.
Thank you. That does add a little bit of the human element. That's what's so hard about some of this. These are black boxes on a screen. But yes, very proud of everybody that's presenting today. This is hard to do. So our next presenter is also a McNair Scholar. This is Stephanie Becklartz. She is in chemistry and medicinal plant chemistry specifically. Her advisor is Dr. Alexander Wilson, and she'll be presenting the effect of foliar applications of methyl jasmonate on the terpene content of cannabis sativa L as quantified by GCMS. And Stephanie, when you're able to, you can bring your slide up and start.
So the effect of foliar applications of methyl jasmonate on the terpene content of cannabis sativa L as quantified by GCMS is really just a fancy way of describing the question, what is going to happen to my cannabis plants if I spray their leaves with hormones? And in this case, the hormone was methyl jasmonate. But really the big question here is, why do we actually care about increasing the terpene content in our cannabis? And if you're not sure of what terpenes are, they're just small volatile molecules that are responsible for the taste and smell of things like lavender or basil or hops, if you're a beer drinker. In terms of industry, they're all over the place. They're in the pharmaceutical industry, the health and wellness, the cosmetic, the food and beverage, et cetera. So those are all billion-dollar industries, so terpenes are pretty relevant. As far as cannabis is concerned, though, terpenes actually help increase the medicinal value of cannabis because of the way that they interact with other molecules like THC or CBD, and that's called the entourage effect. And that's basically the idea that all of these chemical constituents provide a better medicine together than they do as separate individual extracts. So the goal here really is to increase the medicinal value of this cannabis without an accompanying increase in the cost to do so. Methyl jasmonate's not terribly expensive, and it's very active physiologically at really low concentrations, so you don't use that much anyway. So to do this project, I had 4 groups, each with 4 plants, a control group which got no hormones, a group which got 1 millimolar methyl jasmonate, a group which got 1.5 millimolar methyl jasmonate, and then finally a group which got 2 millimolar methyl jasmonate. So the hormone was applied using basically a fancy spray bottle on the tops and bottoms of the leaves. And methyl jasmonate is actually known to regulate pathways which produce terpenes. So the hypothesis for this project was that the plants in the 2 millimolar methyl jasmonate group are going to have an increased terpene content relative to the control in other groups. Unfortunately, my project rather is still in progress, so I don't have direct results. But you can see here on the bottom left of the slide, there's a photo of a plant harvested out of the control group next to a 2 millimolar methyl jasmonate plant. And I'll let you try to make your own conclusions there on if you see a difference. I don't want to bias my research. Currently, what I have left to do, I need to take random samples from each group, take extracts of those samples, and then inject those extracts onto the gas chromatograph mass spectrometer, or GCMS, to analyze the terpene content. And with that, I thank you for listening.
Awesome. Thank you very much. Anyone that wants to, yes. Very good. Thank you very much, Stephanie. We'll let everyone finish their tabulating. And next up, we have Ashley Martinez. She is a McNair Scholar in the History Department. Her advisor is Dr. Robert Goodrich. And today she'll be presenting the culture of remembrance in Austria about the Holocaust. Ashley, when you're able, we'll let you bring yours up and start your talk.
Hi, so my research is on the culture of remembrance in Austria about the Holocaust. So the culture of remembrance refers to the policy of confronting Nazi-era crimes through the frank discussion and by acknowledging the responsibility for the Holocaust. So the Anschluss, or the annexation of Austria, happened on March 12th, 1938. Then towards the end of World War II, a victim theory was adopted by Austria. The victim theory began with the Moscow Declaration that stated that Austria was the first victim of Hitlerite aggression. Then in 1945, the occupation doctrine stated that because Austria's statehood had been dormant during this occupation, they could not be held liable for any of the crimes committed, and therefore they did not have to pay reparations or restitutions to the victims of the Third Reich, since they were also considered victims. However, this Self-sacrificing myth is contradictory to this theory. A major example of this is that while Adolf Hitler entered Vienna in a militarized parade, there were 200,000 Austrians present, welcoming and cheering him on, as you can see in this image. It wasn't until the 1980s that Austria began to fully accept the role that they played in the Holocaust. The Chancellor and President in 1991 gave notable speeches admitting that Austria not only were victims, but also perpetrators. The government began some restitution initiatives. They began putting a World War II memorials along with memorials for the victims of the Holocaust. They began rebuilding synagogues, reopening Jewish museums, and memorializing a concentration camp in Austria that was only 100 miles away from Vienna. So what caused this shift in Austrian government and Austrian policy to adopt a culture of remembrance? Well, ultimately, it was international pressure that stemmed from political scandals that tied Austrian politicians to the Third Reich. And through my research, I examined Austria's culture of remembrance and how it influenced governmental policies, and I highlight the positive and negative changes within the social structure. Thank you.
Thank you very much. Thank you to all of our McNair presenters. That concludes our McNair side of today. So that means we get to do our first People's Choice round. So I will make that available. So on your screen as audience members, you will see an option to choose a People's Choice winner. And we will launch that. Choose that. We'll give it a couple minutes here for everybody to kind of vote and for the judges to finish their thoughts. So we're at 85% of the audience votes. So anyone else who wants to put a vote in, do it pretty soon here, because then we'll continue on. All right. Final call. I'm going to end the poll. So if anyone is still thinking, make your decision now, and then we can get started on the grad side.
Maybe I missed this, but can the audience vote for their favorite McNair Scholar as well as their favorite graduate student, or is it just one across the board?
Oh, yes. Thank you, Emily. You will be able to choose this for just the McNair students. Right now, they have 3 options. And then after the graduate students are done, there'll be another poll for the people's choice just for the graduates. So yes, you'll get a chance for the rest of everyone.
Thank you.
All right, so we're going to end it so we can get started on our next exciting researcher. Like that. All right. So on the graduate side, first up is True Hubbard. True is a graduate student in the biology department, and their advisor is Diana Lafferty, Dr. Diana Lafferty. And they will be presenting Carnivore Guild Structure, the Bobcat's Role. True, when you're able, you can be up on the screen and start your talk.
Hi, everyone. My name's True Hubbard. I'm a second-year master's candidate in the Department of Biology, and I'm excited to be here today to talk about some of my research with you. I've had some amazing opportunities over the past 2 years. One of them has been to be a part of the first ever nationwide camera trapping survey to document wildlife in all 50 states, a project called Snapshot USA. If you've never heard of it, I encourage you to check it out. It included hundreds of researchers across the entire US working together to create this incredible data set that can answer an abundance of ecological questions. As a collaborator on the project, I was super excited to be able to work with this data and answer a question of particular interest to me. My question is, what role does the adorable yet very skilled hunter, the bobcat, have in this diverse US carnivore community? If you don't know, the US has 39 native carnivore species, ranging from larger apex predators such as the cougar and gray wolf to smaller subordinate carnivores such as the red and gray fox, which can be highly influenced by things that occur higher up in the food chain. Bobcats are very adaptable, medium-sized carnivores, also known as mesocarnivores, yet in some environments they have been known to act as the apex or top predator. As such, with access to this incredible nationwide snapshot data, I wanted to examine how bobcat dominant or subordinate status, as well as their daily activity patterns, shift relative to the variation in the carnivore community structure across the US. Using camera traps, a non-invasive method of capturing wildlife images, over 400 bobcat detections were captured at approximately half of the camera arrays located throughout the country. I learned that the highest relative abundance of bobcats occurs in the Southwest region, yet many detections were captured on the East Coast, where the current bobcat population range says they actually don't exist. I found this to be really interesting. Further, of the 9 species for which I had sufficient data for analysis, bobcats were found to have the highest activity level of the 9 different carnivores I compared. This means the bobcat was active for the greatest proportion of a single 24-hour day. Based on the activity curve graph that's located in the top right corner of my slide, we can see that the bobcat, the dark blue line, had a greater density during the daytime hours, and this pattern was consistent across all regions of the US. Lastly, I found that bobcats avoided both coyotes and fox, yet coyotes were actually attracted to bobcats. This may mean bobcats are trying to limit their competition as much as possible, yet coyotes may be trying to take advantage of the bobcat's resources. Ecologically, understanding the carnivore community structure and how it varies across the diverse multi-use lands of the United States is critical to the conservation and management of hundreds of species. The impact of one single carnivore species can cause cascading effects that reverberate throughout the entire food web. As such, we must continue to invest in research that can inform our development and effective management strategies in a world that is ever changing by our human influence. Thanks so much for your time.
Thank you very much. All right. I'll be the first to do the All right, let everyone finish their thoughts and prep the next person. So our next graduate presenter is Casey Juntala. She is a graduate student in clinical molecular genetics, and her advisor is Dr. Matthew Jennings. And today Casey is presenting Are You Infectious? So Casey, take it away.
So it looks like the host stopped my video, but I'll start anyway. So are you infectious? The COVID-19 disease caused by the SARS-CoV-2 virus has affected everyone across the globe and has resulted in over 106 million cases worldwide. A person infected with this virus can display a wide range of symptoms or can be asymptomatic and display no symptoms at all. A PCR test designed by the CDC and emergency released by the FDA has been designated as the standard test used in COVID-19 diagnosis and has aided in performing roughly 312 million tests in the US alone. The sample used for this PCR test is the nasopharyngeal swab, or the little swab that goes up your nose and pokes your brain and generates results within 60 minutes. So what I propose to you today is a fast and simple test for COVID-19 disease screening. The asymptomatic population of individuals that this is targeted for is likely spreading the virus unknowingly and should instead be in quarantine. This test relies on the detection and amplification of 3 specific viral genetic targets, ORF1ab, E, and N, using a technology that is more sensitive than PCR. Using these 3 targets instead of just 1 increases the test sensitivity and specificity and generates results within 30 minutes. Asymptomatic individuals begin the testing process by collecting saliva into a sterile tube. This saliva is then tested using our new gene amplification technique, and results are indicated using a simple color change. So as amplification occurs in the presence of any viral genetic material, byproducts are released, acidifying the solution and causing a color change of a pH indicating dye. So a pink color indicates a negative test and a yellow color indicates a positive test. Using this fast, simple, and less invasive test for COVID-19 disease, screening can be more effective and help people take the proper quarantine measures, further reducing the spread of the virus. In a trial study of 47 NMU campus volunteers, including students, faculty, and staff, were tested using this new technology, and 6 were found to have asymptomatic COVID-19 disease. And to my surprise, my personal sample, which was being used as a negative control, became positive. Additional tests were performed only to confirm I indeed was an asymptomatic individual who the previous day was outside jogging throughout my neighborhood. Lab testing at the NMU Health Center generated negative results, and due to the lack in testing resources, our testing was shut down. The results from our 47 individual trial yielded a positive test rate of 12% as compared to the NMU reported rate of 0.6%. Thank you.
Awesome, thank you so much. So we will, anyone who wants to unmute with me? So some of us are in an office, you can hear distant clapping, although I don't know if it came up on screen. So the next graduate student to present today is Emily Griffith. She's a graduate student in biology department. Her advisor is Dr. Alec Lindsay. And Emily will be presenting sexual dimorphism of long-eared owls, plumage in the ultraviolet spectrum. So Emily, as you're ready, You can load up your stuff.
Thank you for that introduction, Janelle. As she said, my name is Emily Griffith. I am a second-year biology master's student here in the Department of Biology, and I study birds. More particularly, I study bird plumage and the differences between males and females. When most people think about this, they think about birds like cardinals that have really, really bright colored red males and more drab kind of brown females. But some birds and some species like owls, you can't really tell the males from the females just by looking at them. But that doesn't really matter to me because I'm not looking at birds in the visible spectrum. I'm looking at them in a spectrum of light that we can't perceive. So owls, especially the long-eared owl, which is the species that I study, can see in a wavelength of light beyond what humans can see. Humans can only see colors or these different wavelengths in what is called the visible spectrum, but owls can see in what is called the ultraviolet spectrum, which is just kind of out of our reach. Even more interestingly is that if you look at owl feathers under a black light, they will fluoresce a bright pink, which not only makes owls the best birds to party with, but also shows that they have a special type of pigment in their feathers that we necessarily can't see. So what my research does is it looks at these feathers to see if we can individually look at these pigment concentrations to see if there is a difference between the males and the females. So to do this, I've partnered with long-eared owl banders at Whitefish Point Bird Observatory in Paradise, Michigan, which isn't too far from here. And I have taken some feather tips from the feathers they collect while banding to genetically sex the birds. And then I've used the rest of the feather to go ahead and put it into what is called an HPLC, or high performance liquid chromatography machine. This allows me to isolate the particular pigments that cause this fluorescence and measure their quantity by visualizing them through a graph like you see in the bottom there. The higher the peak in the graph, the more fluorescence and the more of these pigments that we can see. And so far, we're finding that males actually do have a higher amount of fluorescence in these feathers than females do, which is super duper interesting. And I am super excited to see what the implications of this research may be on sexual signaling and how we understand plumage coloration moving forward in the future. Thank you.
Thank you very much, Emily. Yes. All right. Thank you everyone so much. This is going so well. I shouldn't say that as I jinx us, but as we toggle back and forth, I think we're doing pretty well for ourselves here. So our next student is Katie Elwell. She's a graduate student in Psychological Science Department, and her advisor is Dr. Joshua Carlson. She will be presenting functional changes in the anxious brain cerebellum following attention bias modification therapy. And Katie, you know what that means. You're on board.
Okay.
The National Institution of Mental Health reported that approximately 68% of US adults experienced high and long sustained levels of anxiety in 2020. This is crazy, but unfortunately not surprising, just considering how awful 2020 was to all of us. Now more than ever, we need an easily accessible therapeutic option for anxiety patients. My thesis, which is a part of an authorized clinical trial funded by the National Institution of Mental Health, is doing just this. I'm investigating two critical components, with one being the development of a new anxiety therapy called attention bias modification, or ABM for short. So this therapy is actually done right on your phone. ABM works by utilizing pairs of threatening and non-threatening stimuli to retrain the brain's dysregulated, hyperactive, and biased attention control to potential threats. So this is a hallmark symptom of anxiety. So because you can customize the pictures and the stimuli in this therapy, it allows one to focus on retraining anxiety specific to the patient. So other therapies are not specific to the patient, they're expensive, they're hard to access, and they have outrageous side effects. However, ABM is affordable, accessible, and can be done right on your phone. Other clinical trials have actually indicated that it is quite effective in decreasing sustained levels of anxiety. I'm also concerned with assessing brain connectivity patterns linked to the cerebellum in anxiety patients. So typically the cerebellum has only been implied in motor movements like walking, posture, balance. So honestly, this is crazy to me because our brain is vastly connected. The cerebellum is a massive structure and I surely thought it was capable of more. So I thought something else had to be going on here. Well, there is. So you see the participants in my study receive an fMRI immediately prior to their therapy, immediately following their therapy, which lasts 6 weeks, and then another 1 month after their therapy ends. So these fMRIs are mapping live moving brain activity via blood flow changes. So this allows us to assess live brain connectivity before and after therapy in these patients. So essentially this is showing us what parts of the brain are dysfunctional and responsible for anxiety. It allows us to map out how one's brain is working. It shows if changes in the brain are long lasting after this therapy. So preliminary data is indicating that the effects of this therapy are long lasting. So lo and behold, there's quite a bit of connections between the cerebellum and threat and anxiety regions in the brain, indicating to us that the cerebellum does in fact play a role in modulating anxiety. Preliminary data indicates that these connections between the cerebellum and anxiety brain regions are decreasing following the conclusion of this therapy, and that this therapy is long sustained. So these findings will have huge implications for both the understanding and treatment of all neuropsychological disorders. I'm excited to contribute to the up and coming literature concerning this new anxiety therapy, as well as contribute to the lack of literature regarding the role of the cerebellum in neuropsychological disorders. Thank you.
Thank you so much. That's great. I love that there's just random people probably sitting alone clapping to no one all over the place. Virtual world is really kind of fun sometimes. So our next graduate student is Emily Klinkman. She is in the exercise science program, and her advisor is Dr. Sarah Breen. She'll be presenting coordination, variability, and injury risk in experienced collegiate dancers. Sorry, my mouth is starting to run out here. All right, Emily, you're next up, so we'll get you set.
I'd like to begin by presenting to you a hypothetical situation. Let's say you're an athlete preparing for a performance. The day before the event, you take a bad landing and you break your foot. You're absolutely devastated and in for a long recovery. Is there anything you could have done to prevent this? Could you have even predicted it? I'd like to explore this situation by focusing on one sport in particular, dance. Dancers are athletic powerhouses who are often under immense pressure to prepare and perform. Injury is all too common in dancers, largely due to the amount of time spent in dance practice, the repetitive jumps and landings, and the hard flooring upon which they spend most of their time. In sports outside of dance, injury has also been linked to a concept called coordination variability. If we break this term down, we know that coordination is how certain joints of the body or limbs synchronize with one another to produce movement. Variability is how much this synchronization of these body parts changes throughout trials of the same task, or the standard deviation. In other sports, literature has demonstrated that coordination variability is higher in a healthy system. It also tends to be higher in more experienced athletes, and lower coordination variability tends to be associated with injury. In dancers, however, Higher dance skill is associated with lower coordination variability, possibly due to the pressure to maintain visually consistent movements. There's limited existing literature connecting injury and dance and coordination variability. So the question becomes, how can we measure coordination variability's relationship with dance injury? So in my study, dancers will come into the lab and do some jump trials in a 3D motion capture field. To calculate coordination variability, we'll take positional data and plot the joints of the lower limbs in relation to one another. In relation to one another in angle-angle diagrams, and the standard deviation is the coordination variability. We will then follow dancers for 4 to 6 months to see how often they are injured. We will set up an equation to link coordination variability, injury, and other factors like how long the dancer has been practicing, how much time per week they are practicing, and how many previous injuries they have had. The goal is to determine the strength of the relationship between coordination variability and injury in dance. We expect to see a strong relationship between low coordination variability and injury, much like what we have seen in other sports. The end goal is to develop a method to improve pre-dance participation screenings. If a dancer has a high risk for sustaining an injury due to their coordination variability, dance instructors could modify training to appropriately minimize injury risk. If successful, findings could also be applied to other sports and be developed into a sport-specific screening protocol. Thank you very much.
Awesome. Thank you, Emily. All right. Thank you very much, everybody, as we're going through these. I'm trying not to race through the next one so people have a chance, but it's kind of exciting to go see what we're going to hear about next. So our next presenter is Social Delgado. She's a graduate student in the Psychological Science Department, and her advisor is Dr. Amber Lacrosse. She'll be presenting fluoxetine as a novel treatment to attenuate cisplatin-induced cognitive impairments. So now she will explain what that means to the rest of us.
Okay. Hi, everybody. Okay, do you know somebody that has had cancer? Do you know somebody that has gone through chemotherapy? You probably do. In January 2019, there were more than 16.6 million Americans alive with a history of cancer. Around 30% of these survivors experienced cancer-related cognitive impairment. So you might be wondering, what is cancer-related cognitive impairment? Some people that have gone through chemotherapy present alterations in certain cognitive domains such as memory, attention, processing speed, and executive function. These are a set of mental skills that include working memory, flexible thinking, and self-control. Alterations in this cognitive domain impact everyday life of cancer survivors, as they report experiencing difficulties in undertaking and accomplishing even simple tasks like keeping track of and paying bills, or even getting ready to go out. As a result, novel therapies are being assessed on their ability to prevent or reduce the effects of these cognitive alterations. My project is interested in fluoxetine, also known as Prozac, a common antidepressant. We want to see if fluoxetine has the ability to attenuate cisplatin-induced cognitive impairment. Cisplatin is a common chemotherapeutic treatment that has been proven to produce cognitive alterations. So why fluoxetine? It has been shown that fluoxetine can produce neurogenesis, the growth of new neurons in the hippocampus, a key area of the brain. It has also been discovered that fluoxetine has anti-tumoral effects. So what am I doing? I started with an in vitro study. This is a fancy word for studies done on cells. I'm treating different cancer cell lines with fluoxetine and cisplatin. I want to determine what's the lowest dose of cisplatin that I can use to kill the most amount of cancer cells. And since fluoxetine has anti-tumoral effects, I want to accompany the cisplatin treatment with this antidepressant so we can not only kill cancer cells, but also have less side effects due to chemotherapy. I'm also doing an in vivo study. This is another fancy word for studies done in animals. I'm treating mice with cisplatin, expecting to see impairment in the mice's ability to perform a behavior task, but also treating them with fluoxetine to see if this drug will attenuate the cognitive impairment. This research has the potential to help better understand the underlying biological mechanisms involved in cognitive decline due to chemotherapy treatment, and will provide us new information on how to improve treatment and quality of life for cancer patients. Thank you.
Thank you. Thank you very much. Mine back on. So everyone, mark down as you need. We'll prepare for our final graduate presentation of the day. So our last graduate student is Olivia Carey. She is in the Master of Fine Arts in Creative Writing program. Her advisor is Dr. Monica McFawn, and she'll be presenting The Sound a Body Makes and Other Ruminations on Navigating Roadkill.
Sorry, lots of clicking. Okay. Hi, my name is Olivia Kingery, and my thesis is a collection of nonfiction poetry and watercolor images, all centered on roadkill. My thesis is a story, so I thought, why not tell a story? But it's not just my story. It's Marquette's story, it's every more than human being's story, and it's our story. I've lived in Marquette for 8 years now. I've watched our shoreline ebb and flow, but the changes that sit hardest are the ones that we don't always see, or the ones that we actively turn away from. This summer, I moved the body of a fawn from the side of Wright Street, just a block from my house. The sky was blue, the sun was shining right there on her, and within the same week, little did I know, I'd be crouched over the body of another neighborhood fawn who had met the same fate. And in my periphery, I can often see people watch what the weirdo on the side of the road is doing. They watch me move deer, squirrels, chipmunks, turtles. And recently, a couple honked and cheered as I was moving the body of a deer from where US 41 and Washington meet. And as I was walking back to my car, all tears and deep breaths, I thought, I hope they would slow down for the next beam they saw across the road. And I hope that my thesis can act as that sort of catalyst too. I hope that the reader can honk and cheer and move on changed, and if not changed, then more aware of the bodies that aren't our own and what they're forced to live through. The images on the screen here are pulled straight from my thesis. The fawn was painted after my encounters this summer. The black lines represent habitat fragmentation, and the abstract red images can be paw prints, blood spots, flower petals. Incorporating watercolor alongside nonfiction and poetry creates a dynamic experience, which is important for a few reasons. No two readers will journey through my collection the same, just like no two beings move along the same path in the same way. A dynamic experience to also showcase how roadkill itself is a dynamic issue. We're not just talking about bodies alongside roads, but intangible impacts that reach three-dimensional into the air, on the surface of the earth, and below the surface as well. We're talking about roadside trash and sparrows' wings adapting to avoid windshields and habitat fragmentation with no animals across the road. My thesis exists because I love the environment. I love Marquette and all the beings that live here. Expressing this love and fighting for the beings that cannot fight for themselves has created a hybrid thesis that tells the story of bodies, bodies of water and bodies of land, human bodies, but more importantly, more than human bodies. The sound of body makes is exactly that. All the sounds that a body can be. Thank you.
Right. Thank you very much. And just a note, Olivia did turn into a dog somewhere in the middle, but that's just because her audio was going out. So we turned off her camera to kind of try to get the internet to catch up, so at least we could hear her.
I'm not a dog, I promise.
If only we could get your mouth to move, you could probably go viral with it. All right, awesome. That is our last graduate presenter. Let me pull this up here. So that means we are up for the next, the last People's Choice round. And we are gonna have, this one's gonna be a little bit more of a break because we're gonna let our judges do their final tallying. We're gonna let the audience do their final People's Choice decision. And then we're gonna do some Q&A and then we'll announce the winner. So stay on to know who won and ask some questions and get to know some of our researchers. But first we'll launch our polls so everybody can vote. All right, there you go. So everyone who was in as an audience member who isn't presenting or judging now has the option to vote. All right. I lost part of my Zoom box, but it turns out I can still do things. So I'm here as a disembodied voice to say that we've ended polling because you all voted. So good job. You all learned pretty quickly for that second one. And now we're going to open up the queue. Oh, there it is. There we go. Thank you. Jeff from AV, by the way, is absolutely wonderful. He's been so helpful getting us to have all this run properly. So we're going to start Q&A. So our very own McNair coordinator, Lindsay Huckla, is going to ask some of the questions that were submitted by audience members. So all the participants, if you can turn on your cameras, we can do kind of a, everyone can see what's going on. And then as she asks the questions, you can just unmute yourself and answer the question if it pertains to you.
Okay, so I'm going to try to keep it in the order you presented in, so it's not so popcorn that we have to be nervous, like back in elementary school reading. So we'll start out with Jalen. Your question is, how are you defining urban in this context, and what specifically are some examples of implementing these efforts in such an environment?
Okay, so first part of the question, how am I defining urban? I would really just say the main part of the research was like city-based. So if it was, for example, like New York City, that's a huge city, that's like a mega city. So it could definitely, this research would definitely apply to that. But then I think that it also could apply to a city in Marquette, for example, that doesn't have a lot of infrastructure yet. So then we could start implementing these like alternative materials and things as a place without infrastructure. So then it's just like building along with that, if that makes sense. And then the second part of the question, could you repeat that, please?
And what specifically are some examples of implementing those efforts in such an environment? So in an urban area.
Okay, so I think first the way to implement it, first off, is just knowing about it. So that's why I'm trying to kind of I like teach that to as many people as possible. I think knowing is the first step. And then after that, it's just being conscious of it when, for example, you're going to build a new house, or if there was like a construction person in this, then they would be like, oh, maybe I should talk to my boss about that. And then like, hopefully those practices would start being implemented over time on like a need basis. So just like, as I need to build my new house, then I will think about using hempcrete over concrete. Or if I'm in a leadership position, maybe, okay, I see that these are maybe a little bit more expensive, but they are better for the environment, then we would switch to that, because without sustaining our environment, then how are we supposed to sustain ourselves, kind of thing.
And then the next question was for Ashley. What is one policy takeaway you think the culture of remembrance in Austria can teach countries who've had a similar history of genocide?
So personally, I think one of the major policies is working with international organizations such as the EU to get policies put across the board. So just how the, I'm trying to think, just how Holocaust denial is illegal in most of the EU countries. This can be put across the board with other various policies if wanted.
And now moving on to the grads. This one is for Casey. Can you state those stats you mentioned at the end, the percentage this method found and NMUs? What are your expectations or thoughts about compliance or engagement with this testing process by the targeted population, since this population seems like it might have an overlap with the population who's unconcerned or skeptical about the virus?
Cool. So the stats that we found in our 47-person trial were 12%. It was 12.7, so 12%. And then the previously reported stat rate by NMU was from winter 21, which was 0.6%, which is an insane difference. And in terms of compliance with this, I would think that it would be a lot better than it is with a nasopharyngeal swab, because I'm sure we've all had that and it is the most uncomfortable thing. And spitting in a tube is quite fast and simple and painless. So even if people have skeptical views of the virus, we can still get more accurate data of who is infected, how it's infecting people, how fast the spread is, and stuff like that. So.
And the next is Katie. And this is a longer one too. All these have been a little more in depth, not the softballs that Janelle was implying that we would be getting. But this is posed as a new treatment for anxiety. Currently, many people have a multi-therapy approach to managing anxiety or their depression, like therapy and medicine at the same time. Did you encounter any data that provided insight into multi-therapy with this method?
Yeah, so attention bias is still in the clinical trial phase, and because of that, our criteria is very strict. Our participants, as well as other studies, they can't have had therapy within the last year, no psychotropic medication within the last 6 months, no neurological disorders, you have to be right-handed, all these crazy controls. And that's just to make sure that the effects that we are seeing are actually from attention bias. However, I do think that coupling attention bias with other therapies will only increase its efficacy. But unfortunately, I shouldn't say unfortunately, just right now it's still in the clinical trial phases, so it's still very controlled. So we are not utilizing it with other therapies at this time, at least.
And then our last question we got in is for Sochi. So chemotherapy, it produces this issue by damaging the brain. Is it known exactly what produces the damage?
Well, with cisplatin, that's a chemotherapeutic treatment that I'm studying. Cisplatin initiates cells death through DNA damage during cell replication and the production of reactive oxygen species, and that impairs the mitochondria. Also, the cell death cause the production of cytokines that pass the blood-brain barrier and damage the neural stem cells.
So let's see if we've had any last minute ones come in. Let's check. Oh, one for Emily about the owls. Can you give an example of a potential impact for this research?
Great question. And I'll go ahead and also preface this with right now, in terms of understanding the differences between males and females, for it really to be shown in the ultraviolet spectrum, it's going to require a ton of data and multiple studies for it to really be understood. But even just these initial steps in understanding how porphyrins are concentrated, if they vary for age or if different types of feathers also vary, could lead a broader understanding as to how we kind of move forward in understanding visual signaling. Some mammals also have this fluorescence to them, which is also currently being studied. It's all a very new and exciting field of research. So right now, any information that we can is going to shape the future.
And it looks like we got one more in for Olivia, if we have time, hopefully. Can you talk a little bit about the process that brought you to this very unique project, your memory about roadkill, or a memory about roadkill?
Yeah, so it's weird. Obviously, people are like, what is she doing? My whole life have felt drawn to the bodies that we've, made natural to see alongside the roads. In my thesis, I talk about being in the car with my mother and my aunt and my cousins when we ran over a family of raccoons. I must have been like 7, but that's the first prominent memory I have. And after that, I've just kind of been led there. I'm wearing these are cat ribs from Roadkill Cat that I got from a roadkill artist. So I've just kind of found comfort in fighting for those bodies that can't fight for themselves.
Very interesting story, and very green, having the recycled materials. So we're out of our questions for Q&A, but there is some still tabulation time. So if anyone has any other questions for presenters, we could do that. Or of course we can all sing the Jeopardy theme song or something to waste time. But otherwise, we're just awaiting a final decision.
Can I ask a question of all the presenters? So I know a lot of you are either undergrad McNair Scholars or graduate students, many of you in master's programs. What are your graduate school plans or professional plans after school, after NMU?
I can go first. Well, I'm hoping to get into a PhD program. I actually have an interview next week. And if I do, I want to become thank you, I'm very nervous. I want to become a pediatric neuropsychologist. So it's just crazy people who diagnose these weird brain disorders.
Nice.
I could go next. So I've currently applied to one grad school, and that is Colorado State University, and they have an integrated resource management program. So basically, that's like fancy talk for just like being a farmer. I just think that would be really cool to grow plants and learn about that process and then make a business out of it. I've been applying to jobs also just in case that falls through. And then I actually, for like housing arrangement plans, I actually am in process of getting a house in Marquette. So I'm really excited about that.
Fantastic.
And I guess I'll piggyback off Jalen. I've lived here for 8 years. I got my undergrad from NMU, not leaving Marquette. My next plan is to have a farm and have some land and then just keep writing about roadkill, eventually get a PhD, but farm is the next plan, definitely.
I can go next. So I'm hoping to get a 1-year internship with hopefully being a curator's assistant. And after that, I would love to get a master's within the cultural studies. And I'm hoping that Washington State is the place I go, but if not, that's okay too.
Wonderful.
I can go next. I applied to PhDs in cognitive neuroscience. I got some interviews and I'm waiting for the response of one program in specific. If not, if I don't get in, I'm going back to Panama to work as a neuropsychologist because I already have a master's as a neuropsychologist. So I'm gonna keep working there.
I can hop on. I recently actually found out that I was admitted to a PhD program at Drexel University. Thanks. So this would be a PhD in environmental science, but it would actually be at the Academy of Natural Sciences, which is a natural history museum in downtown Philadelphia. And my goal and dream would be a museum curator in ornithology. So kind of speaking of roadkill, that's my other role here. I'm assistant curator of ornithology, and I can lead a bird specimen preparation, which is just a fancy word for taxidermy. And I'm really excited to potentially continue that in my PhD.
Fantastic.
I guess I can go. I am hoping to get into human factors engineering PhD programs and work in human exoskeletons. So kind of feedback mechanisms, EMG-based feedback. So that's what I'm hoping to do.
Cool, I guess I'll go. Right now, waiting on PhD school feedback. And then if that doesn't work out, I will resume build and try again next year.
This year has been so hard trying to apply to PhDs. I know you guys all feel me.
And
Yeah.
international student is way worse.
I bet.
So I think, do we have one more still to go?
Maybe. Who haven't we heard?
Stephanie, who may be talking but muted.
Yep, that's exactly what was happening. Yeah, I'm moving for sure. I'm not applying to a grad program right away. I feel like I need, honestly, a break after doing academia in this COVID format. I just want a break from that, but I'm going to move to Washington State. So Ashley, wherever you are, I'll be out there too. And I'll probably apply to actually Colorado State to a horticulture program there, I think. We'll see.
Well, I'm just so delighted to hear all of you present. You're doing just fantastic work. I'm just surprised that the undergraduate research sounded more like graduate work. The master's work sounds like PhD level work. So just keep it up. Keep applying, get in, do us. Oh, and also a plug. University of Louisville has a lot of fantastic programs. We have a really excellent medical school and a lot of really important medical research going on here too. So I know, the cognitive neuroscience and neuropsychology, that there's great work getting done. One of the alumni, Val grad from years back, runs a cancer research center down here at University of Louisville. So there's NMU connections to reach out to and to help you on your journey.
Awesome. Thank you so much, Kristen. Really thank you all the judges for being here. And definitely thank you for taking an interest in all of our research students and what we're all doing. I know from the side of the Graduate Studies Office, we love this event because we hear so much about what's going on other than the administrative side. Like we see forms and we do emails, and it's great to see what's going on kind of on the ground out there. So unless we have any other questions or comments from judges or panelists, we have the scores ready to announce. Ready? All right. So McNair scores first. We have first place is Stephanie Becklards. Congratulations. The runner-up is Ashley Martinez, and the people's choice is Jalen Sims. So if we have enough, yeah.
Woo!
I think everybody has to do the clapping on this thing. It's required. Yeah. So congratulations. We'll contact you. We've got certificates for all of you. There's obviously a monetary prize. And then there's also a digital badge that you can put out on your LinkedIn that says that you participated and that you won. So on to the next round. The graduate winners are Emily Griffith, you came in first place. Social Delgado, you are the runner-up. And Emily Klinkman, you came in as people's choice. So congratulations to her. You all absolutely did wonderful. This went really well, considering this is the first time we've done any of this and there are so many moving pieces. Thank you all for being here and putting in the time. Thank you to our audience for coming and dedicating some time to watching this and for voting in the people's choice. It's really great that we had so much participation. Like, we were worried that people wouldn't bother with the polls, but we actually got 100% in there. So it was absolutely great. And overall, I think it's been an awesome day.
Thank you all for participating. And again, Janelle, thank you. You made it happen. Judges, thank you. Hopefully we see you again next year, and outstanding presentations all around. I learned a ton today. Thank you so much. And I too am proud to be a Wildcat.
All right. Thank you all, and I hope you have a good day. Go out there and think about cool research.
Thank you.
Thank you.